Porque vai. Não, mas tô... que foi? Ah.
Bem, boa tarde a todos. Ana Paula, já conectei lá no YouTube, a hora que vocês quiserem, tá? Já temos 30 pessoas aqui. Se você quiser esperar mais alguns minutinhos, agora fica com você e com a Sara aí, tá bom?
Tá bom, obrigada. Vou esperar dar 2h05 e aí a gente começa, tá? Good afternoon, everyone. Today we invited André Kipnis from Federal University of Goiás, and we are very glad that he accepted our invitation. Thank you, André. And to introduce André, I'm going to invite Sara Rosa, that's our graduate student, to present André Kipnis to us. I hope that we are going to have a very good afternoon of knowledge and discussion.
Good afternoon, everyone. I am Sara Rosa. I am a PhD student of the Tropical Medicine and Public Health Graduate Program at UFG. I am supervised by Professor Ana Paula Junqueira Kipnis and André Kipnis in the project Auxotropic Recombinant Vaccines for Tuberculosis and BCG Revaccination of Healthcare Workers to Protect Against COVID-19. I will introduce the speaker, Dr. André Kipnis, and mediate the discussion. Dr. André Kipnis, graduate in veterinary medicine and animal science from the São Paulo University in 1989, received his master's degree in biological science, molecular biology from the Brazilian University in 1987, and received his PhD in microbiology and immunology from Colorado State University in 2004. He is currently associate professor at the UFG and associate editor of the Journal of Tropical Pathology at Hypothetic UFG. He has a CNPq productivity scholarship since 2006, has experience in microbiology with emphasis on molecular biology, working mainly in the following topics: mycobacteriosis, genetic engineering, infectious bacterial agents, development of recombinant BCG vaccines, and study of iron homeostasis in atypical mycobacteria. At this moment, I give the floor to Dr. André Kipnis. Thank you.
Thank you, Sarah. Can everybody hear me? I hope so. I'm not. Okay, thank you. So I hope everybody's watching, you can see the presentation. So thank you, Sarah, for the introduction. I would like to thank Roberta and Ana Paula for inviting me, and also for this great high-level course, graduate course. And this is the outline of my presentation. As a long-time teacher, I know that presentations shouldn't have too many text, but I apologize in advance because of the students. I think maybe my English or some part of what I say may not be understandable. So I decided to make some slides that are, some of them are quite heavy in text. I'm sorry about that, because I think having the text there, the students can read and maybe understand what I'm saying. Maybe, I hope. So the presentation will be in two parts. One is more a theory lecture class, actually, on iron metabolism from the point of view of the host, mainly in humans, how iron homeostasis is accomplished, and how humans acquire and store and regulate the iron acquisition and availability. Then we'll go on to bacteria in general, and then specifically we'll go to mycobacteria, iron homeostasis, and how these pathogens have the strategies to compete for iron. And in the second part, it's going to be more short. It's going to be shorter. I will talk about some of the data that we have on iron homeostasis in this species of mycobacterium species, which is Mycobacterium abscessus subspecies maciliensis. So this is going to be a different perspective of mycobacterial infection, actually a different perspective of disease development. And I think most of you, if not all of you, know that disease, infectious disease actually, is the result of a different equilibrium between the pathogen and the host, as shown here. And also there's another player, that's the environment, where this relationship is being taken. So there are several factors that will tip this equilibrium toward one side and the other, and will result in either, disease or infection cure or infection eradication. So I think these terms here, probably you all know that, if the pathogen has virulence mechanism, toxicity mechanisms, the infection dose where the infection occurs is also may reflect the progression of disease outcome. And while also drug susceptibility of the pathogen is another important player these days that will favor disease. And from the host point of view, the age of the host may influence the susceptibility or not for infection, for disease development, the genetic predisposition, how this immune response status, also if there is a vaccine for against the pathogen, if there is immunization or not. So all of these aspects will influence the disease development. And from the environment point of view, there are other factors besides these ones that I pointed out, like population density, sanitation status, the preventive measures, and social interventions. But today, what I want to talk about is a competition between pathogen and host from another point of view, which is the competition for iron, which is a metal very important for cell metabolism. And we'll focus this and see what are the determining factors that will favor, in this case, pathogen to establish infection. So iron, as you may know, is a very interesting metal because it can be in two forms. In the ferrous form here, which is this iron plus 2 here, which is soluble. So when iron is in this state, it is soluble, but iron is kind of readily oxidized into the ferric state, which is this other form here, and this is insoluble. And sometimes we might read in some articles that they say they mineralized iron, which is in this form. And I'm going to show you in the next slides why iron is essential for both pathogen and host, mainly because of these two oxidation states it's used. It was the selected metal to be used by most, not all, there are exceptions, but most organisms to perform its functions. And the functions that the iron allows is most of the vital functions of this metabolism cells are dependent on proteins that use iron. Nonetheless, I will show you also that excess of iron is toxic. So cells need iron, but not too much. And I'm gonna come back to this later. And in order to have the right amount of iron, not too much, not too little, there is a tight regulation of the iron acquisition, storage, and availability for both hosts and pathogen. So why is iron important? So iron is present in those two redox forms that I told you, is part of several metalloproteins, those proteins that contain a metal, specifically iron in this case. And those proteins mainly are responsible for oxygen transport, oxygen sensing, electron transfers, catalysis. So here you can see that most proteins, or many proteins of the cells, no matter if it's from the host point of view or from the pathogen's point of view, uses proteins for these functions, and those proteins need iron to have functionality. In the host, in humans and mammalians, most of the cellular iron is in the form of heme, the prosthetic group of proteins, specifically the red cells hemoglobin, but also myoglobin, cytochromes are stored, or not stored, but it's used in this context. But also there's many proteins that have these iron sulfur clusters that also require iron for their function, and so they are the other proteins that also causes the necessity for large amounts of iron. And those proteins are responsible, act as transcriptional regulators and do the catalysis for many enzymes. So I allow myself to use this picture here because everybody's using is many and for other purpose. So the right amount of iron is good, but too much iron is bad. Why is too much iron is bad? Because there is this known phantom reaction. I think many of you have heard of. Actually, there are two reactions here. It's the phantom and this Haber-Weiss reactions. That when you combine those reactions, what you have is that peroxide hydrogen, H2O2, is converted in presence of iron to hydroxyl radicals. Hydroxyl radicals, also known as one of the reactive oxygen intermediates, is highly reactive, as the name is saying. What does that mean? It means that it acts on proteins, on DNA, damaging those molecules. So if these molecules are formed, they are very toxic for no matter what cells. And this happens when there is, especially the ferrous iron, the free iron, soluble iron available. So this is not a molecule, the ferrous iron, this is not a molecule that any cell wants to have it hanging around in the cytoplasm anywhere. So it's either stored in the ferric state or there's no iron at all. Otherwise, these radicals are formed and they will cause lesions to the cells because of the presence of iron. So how is iron acquired in the host, by the host? So it all starts from our diet. We ingest food with iron, and in the intestine, we have iron containing proteins, all different kind of source of nutritional compounds that have iron. And in the enterocytes, in the luminal side of the enterocytes, there are these enzymes that will cause the reduction of the ferric state of iron into the ferrous state. It will solubilize iron. And these proteins, divalent metal transporter 1 or DMT1 proteins that are present on the surface of these cells, but also you will see that in the surface of macrophages, of hepatocytes, and many other cells, they are responsible for internalizing iron intake. This protein was earlier called NRAMP2. So once the iron is inside the cell, it can have different fates depending on the cell necessity. So it can be used directly for cellular utilization to be incorporated in proteins, as I said earlier, the necessary proteins that use iron, or in the case of enterocytes, it can go on to be transported to the circulation, and this is accomplished by another protein called ferroportin, FPN. It's not the same here. So ferroportin is going to transfer the cytoplasmic iron to the circulatory system, which can be distributed to other parts of the host, other cells, other organs of the host, and this is the transportation is accomplished by proteins because the iron is not going to be transported freely. So this iron is taken up by molecules known as transferrins. So then you have transferrins combined to iron, which is then in the circulation taken up by any cells that need it. And this, you'll see how it's taken up. Additionally, iron, when there is excess of iron, can be stored in specific proteins, and one of the most important are the ferritins. So ferritins are proteins specialized in storing excess of iron. So the iron here in the ferrous state is converted into ferric iron and stored in the inside this very complex protein, which actually are composed of 24 subunits of monomers. So as I said, the circulatory iron is mainly coupled with transferrins. So cells that need to acquire iron from the circulation will have transferrin receptors TFR1 or TFR, that will, once bound to a transferrin with iron, will undergo endocytosis, and inside this endosomal occurs acidification that releases ferric iron, and the ferric is converted to ferrous iron by a specific reductase here. And then remember that on the surface of the cells, there is also DTM. So in the endosome here, you have the DTM from the inside to the cytoplasm side here. So it favors the transport of these released ferrous iron from the endosome to the cytoplasm. So in this case, the iron can be used to several needs, and here it's shown that mitochondria is involved in hemibiosynthesis, that important molecule for, myoglobulins and erythrocytes, hemoglobins. And also, just for completion, the transferrin is recycled back to the circulation so that it can take up more iron in a subsequent step. This is more like so that you know how much iron is needed for us. I didn't have this notion of the amount of molecules. So this is one red blood cells, one erythrocyte, and this is the showing the hemoglobin. And inside the hemoglobin, there is the group, the hem group, here attached with an iron. So there are the hemoglobin contains 4 hem groups, and one erythrocyte contains 280 million hemoglobin molecules or proteins, hemoglobins. That means that each red blood cells contain 1 billion atoms of iron. That's a lot of iron, and that's the amount of iron, total iron that we have is about 25 grams, I think, of iron. So, of course, we don't take every day that much of iron. So we recycle iron from hemoglobins constantly. So red blood cells that are dying, that are lysed, or there are phagocytosis, they undergo iron recycling, and macrophages or the reticular endothelial system is specialized on that. So once the red blood cells is internalized, they separate the hem group with the enzyme will release the iron again, the involvement of DTN1, transporting the ferrous iron to the cytoplasm. And again, in these cells, the iron will have different fates, including going back to the circulation by the ferroprotein protein, or stored in ferritins. This is mainly in eukaryotes. The storage of iron in ferritins are mainly in the hepatocytes. But in many cells, all cells have ferritins also. So all of these proteins for iron acquisition must be regulated, because sometimes, depending on the iron availability, the cells or the host will need more iron uptake or less, because if they take too much iron, also there is a problem. So all of the proteins, I just mentioned a couple of them, like ferroprotein, and DTM1, ferritins, all of these proteins expression must be tightly regulated so that when needed, they can be expressed to increase the availability of iron, and also, or repressed to decrease the iron availability. So the host, and as I will show you later, bacteria also have these genes very tightly regulated, and many of these regulations are being learned as we speak. So many of them are just suppositions, and the evidence have made possible for us to understand many things, but there are many others to be understood yet. And one of the main mechanisms of this regulation is by proteins that bind specifically to regions in the DNA responsible for repression or induction of gene expression. Those regions known as operators or regulators which are DNA sequences near the promoter region of those genes. In the case of the host, in eukaryote, in mammalians, there is a systemic hormone, a major systemic hormone responsible for iron intake regulation, which is called hepcidin. This is produced by the liver, and it regulates specifically the ferroportin protein, that protein that's responsible for iron export from the cells to the circulation. So when there is a high level of iron, this is sensed by the liver cells, and it's shown here. I don't know if you can see it well, but high levels of iron is sensed in the hepatocytes, and there is a signal transduction that will induce the expression of hepcidin. Hepcidin is released, it's a systemic hormone, so it will act in different cells. What it does is it induces the ferroportin to be internalized, undergo endocytosis and degradation. So there's those cells that under the influence of hepcidin will decrease the amount of ferroportin. And remember, ferroportin is responsible. Here is an enterocyte. So ferroportin here from the diet is responsible to ingest or to capture iron from the diet to the circulation. Ferroportin here in the macrophage is responsible for recycling iron from a red blood cell, and also ferroportin will be responsible for the iron transfer to the circulation from ferritins, from iron storage. So in the end, there is a decreased iron in the circulation in general because of decrease of absorption, and also because of decrease of recycling of heme molecules as a consequence of hepcidin. And there is also, when is that also needed? Immunity is not very new, it's quite old actually. It was coined because of this regulatory capacity of the host to lower iron levels, especially because when there is an infection or when there's an inflammation, basically, so that the host can control this iron availability or regulate this distribution to avoid the invading pathogens. And so hepcidin was initially, because of that, hepcidin was initially called as a antimicrobial peptide. Of course, it's not antimicrobial because it should have a specific action on microbial or bacteria to cause that, but it's an indirect effect. And so now we know it's not a true antimicrobial peptide. And also, hepcidin is also not only induced, its expression is not only induced when there is a high level of iron. I'm going to go back one slide here to show you. Here, there's another signaling pathway here when there is inflammatory cytokines. Here is shown interleukin 6, that when it's present in an inflammatory state, it also will induce hepcidin expression and secretion and action systemically. So in an inflammatory, in response to an infection, the host will respond systemically to iron lowering levels, as I do, by the way, the mechanisms I already mentioned. Additionally, to a systemic production or systemic action of hepcidin, hepcidin can be produced locally by macrophages and neutrophils. So it can also cause a local a microenvironment, iron lowering levels, not necessarily systemic. So a hypothalamic response, lowering levels of iron, is one of the main mechanisms when in regarding to iron competition, one of the main mechanisms to combat the infection. So one I had already said that the systemic hepcidin response, in this case, when there is an inflammatory reaction by different activation mechanisms, so hepcidin will act systemically, causing lowering levels of iron, but also locally, there are, not locally, but systemically, there are other forms of the inflammatory response cause lowering levels of iron availability that is independent, not dependent on hepcidin. So here is shown that tumor necrosis factor, as well as interferon gamma, will decrease the expression of these molecules. So tumor necrosis factor decrease the expression of that DNT1 molecule, so this will reduce the capability of the cells to intake iron from the diet. Interferon gamma will decrease the expression of ferroportin, so the effect here is similar to hepcidin that causes the degradation of this ferroportin transporter, so that iron is not transferred from the cells to the circulation. And in other situations, the bacteria may be not necessarily in the cytoplasm or extracellular. So in this case here, there is a response to lower the levels in the circulation. So this is a good response to extracellular bacteria. But then when you have a bacteria that's intracellular, like the one that we're going to talk in a little bit, which is mycobacteria, so these mechanisms does not affect much on mycobacteria. So, but there are other ways that the cells may respond to lower the intracellular iron, and it's been shown that the inflammatory cytokines can lower in other ways. For example, the expression of transferrin receptors, which it's a mechanism of acquiring iron. As I said, the transferrin is a molecule that in the circulation carries iron, so we'll reduce the internalization and intake of iron, as well as they may enhance the expression of another transporter or another protein called NRAMP1, and this molecule is expressed in the endosomal compartment, the phagolysosomal compartment. So NRAMP here, when it's induced by for example, for the pathogen, is the way that the whole cell can transfer iron from the phagolysosome compartment to the cytoplasm, and it's good for the whole cell. In this case, if the pathogen is inside this phagosome, like tuberculosis, will have less iron available. So this is another way of avoiding the infection progression, and these are triggered. These mechanisms are triggered by pathogen molecules, pathogen-associated molecules, and pro-inflammatory cytokines. And finally, the host can also express some proteins to help even more to capture iron, and these are, aside from transferrin, these are proteins that are in the mucosal secretions, like lactoferrins, siderocalins, or lipocalins, that are shown here. So these molecules, they have high affinity for iron-containing molecules produced by the bacteria. I will talk about more in a bit, because I'm talking mainly about the host, but bacteria produces a molecule to capture iron, which are called siderophores, which they are the yellow molecules here. So this is bacteria in green. They are producing siderophores to capture iron for themselves. And so neutrophils here are producing these lipocalins that can capture these siderophores instead of the bacteria capturing. So they will have more advantage avoiding the bacteria to capture iron, and eventually will have more success in avoiding the disease progression. So now changing to the other side of this, let's say this tightrope competition for iron, we have the pathogen and focusing on bacteria. So bacteria have many strategies and variations. So it's a very, very vast field here of study depending on the bacteria that you are looking at. And so I try to summarize many of those. It's not as simple as saying that gram-positive have one type, gram-negative has another. So they have combinations, but I will try to make it a little bit more understandable. So what are the strategies for bacteria to acquire iron? So one of the strategies is for them to have proteins that have affinity, direct affinity for these molecules that are responsible for iron transfer in the host. So lactoferrin, transferrin are molecules that contain iron, are from the host, are produced by the host, are in the circulation. So bacteria may have these proteins that have affinity for these molecules. So they are lactoferrin and transferrin receptors that will bind to those molecules and will, inside the cell, will capture the iron that is contained. So it's like a sequestration of the iron from those molecules. Similarly, there are other receptors for those hem or hemoproteins, so proteins that contain these hem group, and and they will also acquire iron by that mechanisms. But one important molecule that is produced by many bacteria, especially those that require iron essentially for its growth and require in an environment that needs to compete for that acquisition, are the siderophores. I don't know if this is pronounced right, but I hope that it's siderophores. And here, the siderophores are these molecules here, shown here in gray. They are molecules that have high affinity for iron, so they can outcompete the iron that is bound. Here, the iron is the red dots inside the molecule. So they can, and this is another from the host, lipokaolin containing iron. So the siderophores will compete and have more affinity for the iron than lactoferrin, transferrin, or lipokaolin. So can gain the access to this iron and complex with iron, and then there is a receptor or some molecules that will combine or will sense and will combine with this siderophore containing iron, and so they can internalize this and use it or store it depending on its necessity. There's an interesting paper about evolution and specifically how this interplay for this competition for iron between hosts, between pathogen and the host, between pathogen and not pathogenic bacteria have evolved, and how these bacteria have established an equilibrium. And you can see that, for example, that between bacteria that are common, that are not pathogenic and pathogenic, there is an interesting competition for iron. So we can give an example of the bacteria that are in our gut, and we have a non-pathogenic Escherichia coli that have a high affinity, that produce siderophores that have evolved to have a higher affinity for iron than other pathogenic bacteria such as Salmonella, Enterica. And because of this higher affinity, these bacteria they can prosperate in the gut and avoid the division or the multiplication or the growth of the pathogenic bacteria. So when we are talking about nowadays more and more about the importance of the microbiome and their role in avoiding infection, and one of the aspects is based on iron competition. So these Escherichia coli can compete for iron and consequently avoid the growth of pathogenic bacteria, and that's why they are probiotic. They're used as probiotics, so we ingest them, and they will avoid the growth of pathogenic bacteria. Also, the evolution of these molecules involved in iron acquisition have caused some bacteria to have specialized host infection. So some bacteria does not infect many hosts, some infect only humans, other infect only other type of mammals. And in some cases, when you look at the iron acquiring molecules, you can see why that happens in part, of course. For example, Staphylococcus aureus, they have a molecule to acquire iron that it binds to lactoferrins from humans with very more strength than it binds to lactoferrin from other host cells. So it have evolved concomitantly with humans so that they specialize in the host that it infects. So the virulence of many bacteria can be related to this evolution of these molecules. Although they are similar in terms of functions, they have very little differences, not little, but small differences that have significant outcomes in terms of the possibility of infection. So here I put some slides about how gram-positive and gram-negative acquire iron. So this is the strategies for iron acquisition. And as I said, bacteria produces siderophores, which are shown here. In the case of gram-positive bacteria, they, as I said, it's not a very split or distinct mechanism. So you will see the same mechanism in gram-positive and gram-negative. So especially because these bacteria can change genetic material very freely by transformation, by conjugation, or transduction. So, but there is let's say, some type of, in terms of teaching or to organize these systems by type of bacteria. So gram-positive have receptors for hemoglobin or hemoproteins, and these receptors in the surface of those bacteria, once attached to these molecules, will be internalized specifically by transporters, ABC transporters, and then the iron is released by enzymes, and the iron that's released inside the cell will be used accordingly. And also gram-positive can produce siderophores, and siderophores, once it binds to iron, it is internalized by specific siderophore receptors. Some bacteria are called cheating bacteria, and for example, they don't produce siderophores, but they have the siderophore receptor, so they can use someone else's siderophore to capture iron for them. It's very interesting when you start to read more about these different strategies for iron acquisition. Gram-negative bacteria, as you all know, gram-negative bacteria have two membranes, the outer membrane and the inner membrane. So it's a little bit more tricky for these molecules to cross both membranes. So usually there are more elements involved. And when we'll come to in a little bit to mycobacteria, and I think you should know by now, if you work with mycobacteria, that mycobacteria is not a gram-negative bacteria, it's a gram-positive bacteria, but doesn't have a membrane, a second membrane. I will come back to this in a bit. So gram-negative bacteria produce siderophores, and also have receptors for molecules, and then these receptors have to use a little bit more complicated mechanism to transport from the outer membrane to the inner membrane, and then use the same ABC transporters so that the iron reaches the inside. And also, gram-negative bacteria have receptors for those transferrins and lactoferrins that we can capture from the host. Usually they are they are extracellular bacteria, so they will have access to these molecules more easily, and then they acquire iron by this pathway. What about the intracellular pathogens in the case of mycobacteria? So they can produce siderophores, and these siderophores will not work on the extracellular environment. They will work. It will depend on where the bacteria is, if it's in the cytoplasm or if it's in the phagosome compartment. So here is shown the siderophores into the cytoplasmic part of the bacteria, of the host cell, to acquire iron, and then eventually be captured back by the bacteria. And also, it can hijack iron from transferrins that are being recycled or that are being internalized by the host cell. So these they can acquire directly from transferrin receptors, although it's not a very frequent source for these bacteria that are intracellular. And also bacteria can escape to the cytoplasm from the phagosomal compartment and can acquire iron that are free, which is not common, but they can also sequester directly from ferritins, which is a very good source of iron in the cytoplasm from the cells. And indirectly, the bacteria can cause mechanisms to increase the iron availability for it. So it can induce the expression of the transferrin receptor. So this will cause more transferrins to be internalized, and you'll have more iron in the cells, as well as it can inhibit the expression of ferroportin. Remember, ferroportin is responsible for iron release from the cytoplasm. So it would inhibiting the expression of ferroportin, it guarantees that iron will stay in the cytoplasm and will have more chance to have access to iron. So, the iron acquisition strategies depending on several factors. I have showed a little bit the differences between gram-negative and gram-positive cells because of their envelope and their proteins. But also, mycobacteria, they have a different envelope both from gram-negative and the common gram-positive bacteria. So they will, depending on this envelope, they will have a particular unique set of proteins to acquire, transport, and store iron. Also, depending on the location of the bacterial infection. So if it's a extracellular pathogen or intracellular pathogen, so if it's intracellular, depending on where it stays in the cytoplasm or phagocytic compartment, again, they will have different strategies to capture this iron. And from the host point of view, it will some organs are more enriched with iron, liver and other organs than others, and also the immune state of the individual. Of course, as I already showed a little bit, how the immune response influences iron availability. So depending on the state of its immune response, will cause different iron availability and necessity for the bacteria to have strategies to acquire iron. If there is if it's in a place where there's a lot of iron, it may require different strategies than in a place where there's very little iron available. So what are the strategies specifically? What are the molecules that mycobacteria have for iron acquisition and storage? So in terms of iron acquisition, the siderophores, mycobacteria produce these three molecules. This is in general mycobacterial species. Again, it's not quite a yes or no division, so I'm just saying for what I say, but mainly mycobactines and carboxymycobactines are siderophores produced by most mycobacteria species, including the slow-growing mycobacteria and tuberculosis here in our example, in our focus. And exocalines is a different type of siderophores that's produced mainly by non-pathogenic mycobacteria. In terms of storage of iron, the ferritins, mycobacteria have produced the bacterioferritins. It's a ferritin that contains a heme group. They produce ferritins, which they do not contain heme group, and I'll come back to this later in a little bit. And also some species produce these molecules called mini ferritins. Are ferritins smaller in terms of quaternary structures? Instead of 24 monomers, they are composed of 12 monomers of proteins, but they have an additional feature that they can bind to DNA. So we'll see, and I will show you later on, that these molecules are important to protect DNA from damage. But this is, and tuberculosis does not have this, the gene for this protein here. So the siderophores that mycobacterium produce, and specifically, I'm now talking about mycobacterium tuberculosis. Let me see my time here. Are two types. The mycobactine, which is a siderophore that's associated to the cell. It remains attached to the surface of the mycobacterium, and carboxymycobactine, which it's secreted. And as you can see here, they are quite similar in structure, except for this acyl chain here for the mycobactine. And this is the part of the molecule responsible for surface attachment. So these are proteins, small peptides, but they are proteins that are non-ribosomal peptide synthase dependent synthesis. What does that mean? That they are peptides that they don't have a gene that codes for these peptides directly. There's not a gene that have an RNA that's translated into this protein or peptide. So how are they built? They are built by many proteins that are the genes of those proteins are represented here. Usually they are named with an MBT letter, and then the number or the letter MBTA, B, C, D, F, J. And each protein of this is responsible for the assembly, and sometimes several proteins together will incorporate one amino acid at a time to make the final siderophore, which is the part of the molecule responsible for attaching iron. Okay, and there are two clusters in mycobacterium tuberculosis, the MBT1 and 2. So the MBT1 cluster have all the genes responsible for the carboxymycobactine synthesis, and then the MBT2 have the genes for the aliphatic, this acyl chain that is assembled in the mycobactine molecules that are going to be on the surface of the bacteria. So remember when I told I didn't say much, but for the host, as in the host, the bacteria, they have to have a very tight regulation on this expression of the genes that are involved in iron acquisition and storage. So this is accomplished by sequences, regulatory systems in front of the genes or in front of the operon. And in case of bacteria and mycobacteria, one of the most important regulators is the iron-dependent regulated box that are shown here, their location on these clusters. So these are DNA sequences. They are next to the promoter region of those genes, and they will be a location where this protein, which is a regulator, binds or not to this region. And once it binds, it can have a positive or a negative effect. It can induce the expression of the genes or can repress the expression of the genes. So there are many different outcomes depending on the genes that they are regulating. And we'll see later on how one example of how this regulation is accomplished. So mycobactines They are produced in the cytoplasm, but they act on the surface of the cell or act extracellular in terms of mycobacteria. So if they are going to act in the cytoplasm or in the phagocytic compartment, they must be secreted. So here is shown the molecules that have been recently, not so recently, but in the last 20 years or so, shown to be involved in mycobactine secretion or translocation. So you have molecules involved in mycobactine translocation to the outer part of the cell or secreted, and other type of proteins for the internalization. I must have a quick stop here to say something, because I'm a teacher, and as a microbiology teacher, and as I already said, mycobacteria is not a gram-positive, it's not a gram-negative bacteria, it's a gram-positive. And I came across this figure, but I didn't find another one to put in this place. So I apologize for part of this figure. I hope you know what I'm saying, but you know, mycobacteria, they are, as gram-positive, they don't have an outer membrane, although many papers recently say they consider the mycolic acids, the large amount of mycolic acid and lipids that they have on the outside part of the peptidoglycan as a membrane, because they are so rich they're so hydrophobic that in the end, it's very hard for compounds to get into the cell because of this large amount of lipids. But that's not a membrane. But people call it outer membrane. Okay, but to draw a schematic diagram of the outer side of the mycobacteria like this, and saying this is an outer membrane, I think this is a little bit wrong. So I'm sorry if anybody saw that before as I was talking. So this is not how a mycobacterium have organized the mycolic acids in a lipid bilayer, but it's how they represented. And as I said, I didn't find another picture. But the effect is kind of the same. So the molecules, specifically the siderophores, that needs to be secreted or internalized, they must cross this mycolic layer on the outside, and then they must cross the inner cytoplasmic membrane. So having said that there is I think it's an error to represent this envelope of mycobacteria like this. But let's assume that this is the mycolic layer. So once the siderophores are synthesized, it doesn't matter if it's carboxymycobactine, which is represented in this green without this tail, or the mycobactines, they are secreted by these proteins that they are called MMPL or MMPS. So these are a group of proteins that are called mycobacteria membrane proteins, large or small. Usually they are in pairs, the large with the small, and there are many, and they are kind of specific for mycobacteria, as the name says it, and they are usually they are very similar, or they have a function of efflux pumps, and some of them are involved even in efflux of antibiotics. So in antimicrobial resistance, that they pump the antimicrobials to the outside of the cell and avoid creating a concentration inside the cell, and they become resistant. So these are the MMPLs that are involved in siderophores secretion, the MMPL 5 and 4, and their counterparts MMPS 5 and 4. And so they are secreted. The mycobactines remain attached to the surface of the outer layer of the mycobacterium, and the carboxymycobactines are secreted, and they will eventually compete for free iron where they are. And once they acquire this iron, they will transfer. It's not quite exactly clear how they transfer this iron to the mycobactines. The mycobactines will internalize this iron, and then there's the role of these proteins FECB or FCB2, which will transfer the iron to the transporter of iron to the cytoplasm, the IRTA and IRTB transporters. These are molecules that can transport the molecules, the siderophores, into the cytoplasm. They also have in their complex reductase activity necessary for iron release and then posterior use. I put here this important, interesting observation that the mutant, the double mutant for MMPL and MMPL4 and 5 is the strongest, is the mutant that had the strongest attenuation among the many mutants that have been done in genes related to iron acquisition and storage. So when you have a strong attenuation, so there is no redundancy, other proteins that can take place when they are not present. So that means that these are good targets to develop drugs against mycobacteria, because if they are almost like essential for infection, these are good drug targets to think about. And also, there's also another group of proteins that are the secretion type 7 proteins. I don't know if you're familiar with that. I'm not going to talk much about it, but the secretion type 7 family of proteins, they are called ESX 1, 2, 3, 4, 5. So the ESX1, again, if you are in the mycobacterial field, you should know that ESX1 is responsible for the export of CFP10 and SAT6, while ESX3 system is important for iron uptake. So it's a specific group of secretion system that require more attention, and it's being studied extensively to see how exactly it's involved in this acquisition of iron and maybe target for drug development. So until recently, they thought that mycobacteria did not acquire iron from a hemp source, but more recently they have shown that there are proteins on the surface of the bacteria that also can bind to heme, and so the studies have shown that they are involved in heme binding and in internalization. So these are the proteins that they have associated with this binding and internalization, the RV0203, and then you have the efflux pumps involved with that internalization, composed by NMPL11 and 3, and another group of proteins which are from the family of the PPE proteins, proline glutamate proteins that are also involved with, seems to be involved with that mechanism of iron uptake. And they use, in this case, they identify the involvement for the heme intake, the involvement of this protein, which I just showed you that it was also involved in the intake of siderophores. So there seems to be an important role for this protein, which again is an interesting target for drug development. So how are those genes, or some of those genes? I don't have time to go much further, but how those genes are regulated in terms of expressions, and using the siderophore synthesis as an example. I told you that the genes for siderophore synthesis, they have sequences that bind to the repressor. In the case, it's the IDE, iron-dependent repressor, which is a protein that binds to iron. And when it binds to iron, this is the repressor bound to iron, it will bind to the box or to the DNA sequence, and once it's there, it will repress the expression of the siderophore synthesis, right? It makes sense. There's a lot of iron in the cell. The mycobacteria does not need to acquire more iron, so it needs to shut down the expression of the genes responsible for siderophore synthesis. So this is the repressor, how it binds. This is, I think it's, the behavior here is just like the lactose operon. Even the induction is, I think it's quite similar. So this will be repressed by the presence of iron. And when there is no iron, theoretically, no iron, the repressor will not bind to the IDR regulation box here. So no repressor present. There should be expression, right? Okay, but it's not enough. So in this case, there is a second stage regulation. So there is the need for a positive regulator. There is the need for the binding in this region for the molecule called HUB-B. So it's another molecule producing another region of the genome of the tuberculosis, the HUB-B. So just the absence of the repressor here is not enough for the expression of the siderophores, or the proteins responsible for the siderophore synthesis. It requires a positive regulator. So if it's not clear how this is working, I suggest you read more about the lactose operon regulation, which is also have a positive regulation made by AMP, cyclic AMP. So mycobacterium produce and tuberculosis produce those siderophores, and then once it acquires iron, how it's stored, where is it stored? So in tuberculosis store have two genes coding for ferritins. One is called the bacterioferritin, BFRA. As I said, it's a ferritin that has a hem group, and also another gene coding for a similar but different, a different ferritin, which does not have the hem group, the BFRB. And it was shown also that more recently that although they thought they were redundant, actually they are not. They're especially important for during infection, and they are involved. So the ferritins there is expressed differently, especially during infection, and that is related to iron availability. So basically, the bacterioferritins are more important, are more expressed during low iron conditions, as those mimicking by when there is a cause during infection inside the macrophage, as the cell will try to lower the iron levels available. And when there is a high amount of iron available, it will require the expression of both ferritins so that it can have store this excess iron and avoid the toxic effects of excess iron. So to end this part of my presentation, these are some molecules or some compounds that already are available or are being studied that will act specifically in this metabolism of iron and have effect on mycobacteria or have anti-mycobacterial effect. One of these compounds is a very old drug used for the treatment of tuberculosis. If you read the old text, you'll see that prior to isoniazid and rifampicin, they used to treat patients with p-aminosalicylic acid, and of course, it's not very as effective as those other drugs, but they served their purpose when there was only this kind of drug, and now they know that they target mycobactine synthesis. Another strategy is to use siderophore analogs for siderophores, in other words, drugs that can chelate iron from the siderophores, from the mycobacterial siderophores. So one of the drugs is PZP, which was shown to have a good antimycobacterial activity. And the drugs that are targeted to those efflux pumps like MPL3 are these compounds, and also some adamantyl-urea-based compounds that were shown to have antimycobacterial activity by inhibiting specifically the heme uptake. And again, I told you about this model, this protein as a target that is being studied, but I didn't find any compound specifically that will bind to this molecule. So I don't know if you want us to have a small break here, or we should continue. I think I have 30 minutes more at most talk.
I think maybe it's an opportunity for everyone to think over if they could understand what's the mechanism involved in iron acquisition, and for some questions before you continue. So it's open for questions for this part of the presentation.
Is there any written question? I don't see that.
Hi, André, it's Milton here.
Hi,
can I go first?
Sure.
So thanks very much for a very nice talk so far, and a very interesting topic. I believe that nutritional immunity is one of those issues that has been raising a lot of attention in the past, let's say maybe 5 to 10 years, right? And well, a lot of new genes is being described and associated with either regulation of different elements like iron, or even some amino acids, right? And this is important because maybe we then have a link how did those networks are actually associated with what we call the social determinants of health, right? I mean, if you take tuberculosis, for example, we understand that obviously there are a lot of the different aspects and factors influencing the outcome of disease. But what we understand is that obviously the social determinants of health, which is very difficult to define, right? But it's shortage of food, or environmental conditions, and household conditions, and things like that, which is obviously associated with extreme poverty in most of the times, most of the cases, right? So anyways, we understand that if we also taking into account that we are talking about chronic diseases such as tuberculosis or leprosy, we understand that sometimes we cannot actually have a very strong association. For example, if you take vitamin D and levels and in leprosy patients, and this is data from Roberta, and we do not see actually a deficiency in vitamin D among patients. So it's not and obviously always all the patients will exhibit some sort of anemia, I would say, right? Because this kind of battling between bacteria and the host towards iron, for example, leads to exhaustion of this element in the circulation, right? And so it's more like a comment, but I'd like if you have any idea of papers that actually can associate all of those conditions that we call the social determinants of health and the outcome of those diseases, and which would be the contributing factors towards these the most important ones, at least, that could go towards the outcome of disease.
Okay, Milton. So it's not a question. I know because it's very hard for us to think about it, because first of all, I think this is a multifactorial component. This is response, and I don't know if I could show you, or if you, of the people who were listening, understood that there are several ways to accomplish the same thing, several ways to acquire iron, several ways to store, and several different strategies when we look in one particular organism. So these are redundant conditions. One of the things that it shows me is that it's very important. So that's why there are several. So the bacteria cannot afford for a mutation or a deletion of a gene that is essential, because otherwise it would die. So that's why they have different mechanisms to iron acquisition and storage. And one of the interesting books that I read that that made me think about this this aspect of social determinants. I don't know if you read it. It's the survival of the sickest, and it goes on to tell the story of the how disease have killed many people, especially the the Black Plague, and the outcome of that. There are some suppositions, for example, that when you look in the European population, they have a mutation that in many people, it's a high incidence of a mutation in a gene that causes hemochromatosis, which is a disease that causes high iron in the circulation. And he goes on, but although there's high iron circulation, the macrophages of those individuals, they have lower iron levels than normal. And so they go on. The book is fictional, but it's also has a lot of a scientific background. So they kind of go on to the theory that those individuals survived the plague because of that mutation, caused this ability for the macrophages to deal with the infection, with the Yersinia pestis infection better than those that didn't have the mutation. So when you look at this evolution in the long term, maybe you can see some aspects of the importance of some genes or some players that are really necessary, but it's very hard to see effects, like you said, when you treat with vitamin D. Also, the NRAMP gene, NRAMP1 mutation was first thought that this was really a marker of resistance, but then they went on to see that it's not clear cut, especially I think because there are several strategies that circumvent a specific maybe something that could favor one side or the other. So it's very hard to study these metabolisms and see their really importance in disease development. I don't know if I
yeah, that's right.
help.
Yeah, thanks. That's the point. And if I could just say, I don't know, Alberta, if is there any and Ana Paula, if there are any other questions, but just to mention that we are investigating in leprosy at least some of those genes associated with the disease outcome, and one of them is pyruvate kinase, and which is actually selected. We understand that is a selective pressure in the PKLR gene in Africa for malaria, and which is the resistance that we see in African populations is obviously associated with phenotypes that are especially in red cells. I mean, the phenotypes have decreased half-lives, for example, for the red cells, which is very, very interesting, because then the plasmodium won't live inside the red cells, right? But anyways, this also means that you have increased iron concentration in the serum of those individuals, right? Which means that this iron will end up in macrophages, and then they can have increased susceptibility to mycobacterial diseases. And we are about to publish this findings. It was a matter of a PhD thesis in my lab. And actually, I mean, this what we are calling a kind of a trade-off evolution, right? Which is can be advantage in one hand because we can see for malaria, and on the other hand, we can see a disadvantage for mycobacterial diseases. And these are the kind of what we call the, genetic footprints that are we can observe in some specific populations. But I mean, this is a very, very interesting discussion, and thanks again. I'm sorry I took a lot of time, Roberta. Maybe somebody else can ask questions.
So just to comment, this trade-off, when you get resistance to one infection, like you said, malaria, and susceptibility to others, it's a lot discussed in the hypothesis of the, if I'm not wrong, Red Queen hypothesis of the Alice's in Wonderland, where the bacteria and the host are mutating, are constantly changing, and they are, evolving to avoid one another, but then they are in the same place. So they may gain advantage for one thing, but then they, the fitness for other infections is not so good. So
yeah, it's really amazing. It's great.
Professor Yerny Prado has one question. Yerny Prado said, excellent presentation, Dr. Kipnis. I have question. The pathogenic and non-pathogenic mycobacteria use the same regulatory process for ion uptake.
Yes. The question is depending on the bacteria that you look into. So they may have sometimes, and when you say they have the same regulatory process, they may have the same tools, the same molecules, like they both produce siderophores, but then it's not just the siderophore production. You have to they investigate better what's the affinity of those siderophores. For example, if they are chelating iron from a transferrin or from a lactoferrin, sometimes the siderophore from the pathogen is it can bind iron from the lactoferrin than the siderophore from the non-pathogenic bacteria, or vice versa, as I said in the example for the gut bacteria. So sometimes they use the same strategies, but they have differences, little differences in the affinity for their ligands. So that's why sometimes just looking if they do have or not is not enough. You have to measure their characteristics in terms of the affinity.
Thank you.
And
I have a question. If you guys so you can tell me
and also
when I can ask something here.
André, you can ask him now. André Bafka, please.
Hi, Shara.
Thanks,
André, for the brilliant talk so far. And so I'm really curious to know whether the MTB deals with iron while it's outside of the cell. So in that step, when it reaches tissues and is outside, so if there's anything, it's known that is something that is taken up from the transferring. I'm very curious about that.
I haven't read any specific approach that can deal with iron acquisition during infection when the bacteria is extracellular. I think most of the data, it does not make this distinction. It's infects and it doesn't. It's very hard to compartmentalize. So you can investigate the bacteria, how it's acquiring iron outside the intracellular, environment. So I'm not sure. And I have here Fabio Muniz, which is, I will present to you later, but he's done most of the work here. I don't know if he has any input into that. So I'm not sure that. So as I said, there is a way. There is a immunoregulatory response to induce transferrin receptor increase. So, but that's for the intracellular condition, so that it can have more iron in intracellular. But mycobacteria extracellular, I haven't read anything, but I may have missed that.
Thanks, André.
Hannah Prata said, interestingly, I also saw an increase in cytoplasmic HER1 in mycobacterial avian infection. I haven't tested iron yet, but it could possibly be the same mechanism, right?
Hannah, I think you have to, you have to see the way to measure iron contents inside. Yes. I also, in my view, I haven't gone into that too much. So Fabio Muniz, Fabio, do you want to talk a little bit? I think he doesn't want to speak English, but I think he can have some input into this question.
Hi, I'm going to try to talk English and try to say something about some questions and about the how the mycobacteria regulation process. The both mycobacteria have the same process when they think about the regulations, process regulation, high the air and the hook B. So is the process involving intake and the storage of the iron. When the Professor André Kipnis asked about how the cells mycobacteria is going to get iron outside of cells. Sorry, I think my microphone is not good.
We are listening.
Are you listening?
Yes.
And yep. the main source is ferritin. The professor there is going to explain more about that, but as I showed, during infection, the initial stage of infection, mycobacteria use the iron storage. So the iron is not during the outside cells. The cell, the mycobacteria uses the iron inside of the cell storage in ferritin. So the main source of iron is an iron inside ferritin. So the other mechanism like Siderov and the orders like the AMI is not very used because the cell during the first initial infection, the immune nutrition is going to reduce the iron, and so the cell have to infect the host cells to initiate the infection. So the ferritin is the principal source. I don't know if you understand.
Yes, we did. Thank you, Fabio.
Thank you, Fabio.
Sarah, do we have more questions?
No, there aren't.
Okay. So let's go on now for the second part, okay? As I said, I shouldn't take long. So the second part is about the work we have done with this species of bacteria, which is Mycobacterium abscessus subspecies maciliense. This is the most current nomenclature for this bacteria. This is a fairly new species, at least it was reported. I think it existed for a long time,
André,
but only, yes.
Nós não estamos vendo a sua apresentação.
No, sorry. No, again. What about now?
No.
Just a second.
Now we are seeing.
Okay, sorry about that. So the name of the bacteria is Mycobacterium abscessus subspecies maciliense. So as I was saying, it was reported in the in the beginning of this century, but I think it existed long before. It's just that they sequenced the whole genome and realized it was a different species from M. abscessus, Mycobacterium abscessus. So just a second again. Okay. This bacteria became important in 2005 and 6, especially in Brazil, when an important infection outbreak occurred. So this bacteria is associated with infections in cystic fibrosis patients that have problem in the surfactants of the lungs, but also is very much associated with post-surgical and post-traumatic infections. The post-surgical infections is mainly due to problems with the disinfection and the sterilizing of the equipment used for those surgeries, and they are usually associated with videoscopic surgeries. And in the case of the outbreak that happened in Brazil in 2006 and 8, around 2006 and 8, it was specifically, especially in Goiânia, at least I can say with more precision, it was associated with the surgery of the knees, of the knee joints by videoscopy, and it took quite a while to realize that it was a bacteria infection, because the individuals had their surgery and they only became with symptoms 6 months, 8 months later, and then took a while for them to realize that it wasn't the reaction to the suture of the infection, but the suture of the surgery, but to a bacteria. And because this bacteria takes a while to grow in the laboratory, not as much as tuberculosis, but it does take, so it's more than usual, and people were missing that in the laboratory. And here's a map of the places where this outbreak occurred in several states of Brazil, Belém, the city of Belém, Goiânia, Vitória, Rio de Janeiro, São Paulo had a city with high incidence, and in the south too. When we got a hold of this bacteria, others showed that in Sylvia's group that they were they had some resistance to glutaraldehyde. Glutaraldehyde was one of the sterilizing agents used to sterilize the equipments, and they were doing it properly. So the surgeons were transmitting the mycobacterium to other patients when they were doing their surgeries. In addition, this bacteria, this group of bacteria, not only maciliense, they are resistant to many antimicrobial drugs, in particular the anti-tuberculosis drugs. So in the beginning, they even started treating these patients when they realized they were infected with this bacteria. They started to treat with anti-tuberculosis drugs like isoniazid, rifampicin, all the drugs which are very toxic, and they have no effect on these bacteria. And so there are a few drugs that are active against them, and we have shown with others that this bacteria seems to be more virulent than the M. abscessus subspecies abscessus. And again, maybe going into where André was asking, this bacteria, when it infects the host, although it does induce some granuloma formation, it seems to be more extracellular, induce more lysis of the host cells and being extracellular compartment than tuberculosis. So we had these bacteria with an interesting virulence characteristics, and we sequenced the whole genome of one isolate at that time, and later we started to ask questions that about their iron homeostasis mechanism. So we reasoned that they should have different iron storage mechanisms because they have a different pathogenesis and mechanisms of inducing the disease. So we went ahead and asked what are the strategies that these bacteria use for iron storage, and also are there other roles of those elements in virulence? If they are similar or different, if they are different, is there a consequence in the virulence? So the first thing was to one of the first things that we addressed was does sorry about this writing here. Does this species of bacteria have the gene coding for bacterioferritin, or the similar gene of M. tuberculosis BFRA bacterioferritin? So we did this in silico analysis, and it came out that the group, all the three species of the M. abscessus group, M. maciliensis, M. abscessus subspecies abscessus, and the Bulletti group, none of them had gene coding for a similar protein as BFRA. And in regarding to the other ferritin gene, which is the BFRB, the ferritin gene of tuberculosis, we found that the M. abscessus subspecies maciliensis, or MICMA, I will call it MICMA as short for M. abscessus subspecies maciliensis, they had two genes coding for a protein with around 70% of similarity to the BFRB. They were not identical between themselves. They were also they had high similarity, but they were not identical. They had higher than 70% similarity between them, but also between them and the BFRB, they had this similarity. So the next other question that we went further was to see if they would be they are really true to being expressed, and also if they had function as a ferritin as predicted. I will here, this is just to show that the ferritin, this is the MICMA, the 0077 gene is here, and it's quite different from the MICMA 0076, the other ferritin gene in M. abscessus subspecies maciliensis. So first we did some analysis to see if the growth, in vitro growth of the bacteria was affected by iron availability. So they didn't have one of the ferritin, the bacterioferritin gene had two genes for ferritins. So did that imply any maybe in problems in growth according to iron availability? So here we have media without supplementation of iron, but that's usually these media have some iron, residual iron left. and the bacteria grew well as when there is sufficient iron or even excess iron. This mycobacteria grew well in the presence of different concentrations of iron, so tolerated high levels of iron as well as very low levels of iron. Only when you retrieve or when you remove total iron from the media, then you see the bacteria suffering to grow. And we analyze a different iron availability, disponibility, if the expression of if those genes were expressed, and also if they were differently expressed according to iron availability. So we analyze both genes, 0076 and 0077. This is a relative gene expression. That means that it doesn't mean that there is no expression of gene here. It's just that we normalize the lowest expressed gene as 1 and all the other relatives to this condition. So that you don't say, oh, there's no expression here. There is both genes are expressed. So the short here is that both genes are, let's say, functional, because we detect RNA of those genes, of both genes, but they are expressed in different levels. Different levels in terms of even when there is no iron present. And this one, in the case of the 0076, it does have a positive correlation in terms of iron availability. So the function of this gene, again, is to store iron, right? So when there is a more availability of iron, the 0076 is in vitro, is induced so that probably can accommodate this large amount of iron and store. And this is our hypothesis that in excess of iron, the 0076 is the most important one for iron storage. We went ahead and did the how those genes would be expressed during the macrophage infection, and here we see different. We see that both genes are induced during a macrophage infection. But the 0076 gene is peaked. There is a peak of expression 24 hours later, but then it's suppressed. While the 0077, there's a little bit different behavior that remains expressed until 48 hours. And this is not correlated with the increase of mycobacteria inside the macrophage. It remains, although there's a difference from the initial infection, but there's not a very exponential growth inside macrophages. And another observation that we did see is that the infected macrophages, as the infection progresses, you can see a large amount of extracellular mycobacteria, and this is not a result of extracellular mycobacteria multiplying, because the media was changed. So there was lysis of the mycobacteria to the extracellular, and so we conclude here that there is a different pattern, probably reflecting different roles of these genes during macrophage infection. And the mycobacterium does gain access to extracellular space, probably by macrophage death or by apoptosis. So to characterize those proteins, not only to see if they are expressed, but to see if they are true ferritin, if they behave like ferritin molecules, we cloned those genes, both the 0076 and 0077. We got more data from the 0076. So we cloned, expressed, purified, produced antibodies, and then we went on to see the location, that it's not secreted, it's not in the supernatant of the growing bacteria, but it's in the, we cannot locate specifically, but it's cellular, it is in the cellular fraction. You cannot say it's in the cytoplasm, but it's probably in the cytoplasm of the bacteria. We also produce the bacteria, the protein in the native form. so that we could do a non-denaturing gel and show that they form high weight oligomers. And when we purified in filtration columns, we could determine the size, the expected size was compatible with the formation of those 24 monomers that are characteristics of the ferritins. So they are expressed, when expressed, they form these oligomers, and then the other question is, do they have the activity to oxidize iron? So I may have missed this explanation, but not only the ferritins store iron, but in order to store iron, it must take the free iron in the ferrous state and oxidize it into the ferric state. So the capability of the protein to oxidize iron from the ferrous to ferric state it's a necessary step in order to store it. So this is the assay to see that. So when the assay, when there is a ferrous complex with ferrocene, you will see a color production that can be measured in absorbance. And when you combine the ferrous and the recombinant ferritin, or the recombinant 0076 protein, in the presence of ferrocene, you see that there's no more production of color. There's no more ferrous iron available. There's only ferric state. So the color changes from this state to this, meaning that as time progresses, the recombinant protein can oxidize ferrous into ferric form, and this was done for both 0076, recombinant 0076 and 0077. So the other question that we wanted to see if we could answer is, since this bacteria does not have the bacterial ferritin gene, it's absent. Roles of this gene in virulence, because if it is present in the pathogenic Mycobacterium tuberculosis and is absent in this not so much pathogenic Mycobacteria, does that mean that when you insert this gene and you have a Mycobacterium abscess expressing this protein, does it confer any advantage? So in order to do that, we did a recombinant Mycobacterium abscessus maciliensis. This is not by recombination. This is only expression in the episomal, in the plasmid. So this is just confirmation that the Mycobacterium abscessus subspecies maciliensis containing the plasmid does express the bacterial ferritin, although there was some very cross reaction with the ferritin itself. And we analyze how these recombinant bacteria, Mycobacterium micma, would behave in the presence of higher levels of iron, as well as in the presence of oxidative conditions like presence of H2O2 that predispose the phantom reaction, and in the presence of iron, so it can be deleterious for the bacterial growth. And we did see in all the conditions here shown in different concentrations of iron, as well as in the presence of hydroxy oxygen, it's the recombinant bacteria, which is the black or the gray curve here, they always could grow better than the wild type, the micma with the empty plasmid, with no expression of the recombinant BFRA, or the bacterial ferritin. So they showed superior growth in iron and oxidative conditions. So we got excited with that result, and we thought that we would have very good differences in infection of macrophages as well as in infection of mouse. So we first infected this lineage of lung macrophages, and we didn't see any difference in the CFU during for doing some 72 hours of infection. So they behave similarly. And in the case of M. abscessus subspecies massiliense, the wild type mice is more resistant to infection, so you don't have a good model of infection. So we use a more susceptible mouse, which is the interferon gamma knockout mice. They will develop more infection. And what we did see was a difference in terms of inflammatory response, no different number of bacteria recovered from those mice, but we did see, and in the right side here are the lungs of mice infected with the recombinant MICMA expressing bacterioferritin. So I think you can compare with the left side here. This lung here, although there is some inflammation, it's less so than the mouse infected with the recombinant bacterioferritin expressing MICMA. And here we show more in detail this inflammation characteristics. So the recombinant MICMA does induce more inflammation. And in the case of MICMA, we also saw that they do have, as expected, another ferritin in their genome, which is a ferritin, the mini ferritin, which I don't think I think you remember I talked about it. It's called DNA binding protein DPS. It's a ferritin molecule that forms in a smaller number of monomers instead of 24 or 12. And but in addition, they have a capability to bind DNA. And the idea here is that these proteins, they will, by binding or staying close to the DNA and having the ability to oxidize the iron, it protects the DNA from free iron and from the phantom reaction that may develop near the DNA and consequently damage the DNA. So we did a similar approach with this gene. We cloned the gene with similarity to DPS and expressed the protein purified, and the nature protein that was produced formed, again, oligomeric complexes compatible with the 12-mer structure in non-denaturing DNA. And the assay that we performed is protection of the DNA by two aggressive mechanisms. So first here, with the DNA here that we are using is the plasmid DNA. So we make a system of the plasmid DNA with the enzyme, in the case DNase. That's all you know, is an enzyme that degrades DNA. So here is the control, and when you incubate that with DNase, you don't see the DNase anymore. So as you know, plasmid DNA has two bands. But if you incubate plasmid DNA in the presence of the DPS, of the recombinant DPS protein from MICMA, what you see is that the DNA remains intact. There is a change in the conformation of the plasmids, but that's also reported by others that by binding to DNA, it changes the supercoiling effect of the plasmids. But most importantly, the recombinant protein protects the DNA's activity. So this is first an demonstration that by somehow the DPS will be around the DNA or bind to the DNA and avoid the contact of the DNase to the DNA and degrade it. So, but the actual effect of the protective effect of the DPS protein is by avoiding the phantom reaction that in turn will degrade DNA. So in order to mimic a more physiological activity, we did a similar assay, but instead of incubating with the DNase, we incubated the plasmid DNA with hydrogen peroxide in the presence of iron, so that the phantom reaction can be developed, and then reactive hydrogens can be formed, and this is the control without hydrogen peroxide, where you have the plasmids, and then we skip this one, and you see that in the presence of hydrogen peroxide and in the presence of the protein, you see that the preservation of the protein. And now here that I'm noticing that this is wrong, Fabio. Sorry about that. While in the presence of hydrogen peroxide, in the absence of the recombinant protein, the DNA is quite degraded. You don't see as much intact DNA. So again, this is a more physiological assay to show that the lesion caused by hydrogen peroxide on the DNA is preserved when you have the TPS protein in the incubation. So, as shown, we have 2 genes coding for ferritins in Mycobacterium abscessus subspecies massiliense, with different roles in the infection of macrophages and possibly in the infection of the mouse. When we express the recombinant bacterioferritin, it does confer advantage for toxic survival. The absence of this bacterioferritin and the presence of 2 ferritins may be a reflection of the evolution adaptation of this bacteria for infection. And we also show that these mycobacteria have mini ferritin or a DPS protein that can protect DNA from damage. So although we did show these data, the next step, which is being done as we speak, actually, is quite, there are many data now, is that we did knock down one gene, one of those ferritin genes, the mycoma 0076 from the Mycobacterium abscessus subspecies massiliense, and now we have the recombinant mycobacteria, and we are investigating what's the effect of the absence of this gene. So I think we can have more insight into the necessity or the viability of the cells and what is the role of those recombinant mycobacteria during infection. So this is part of the postdoc of Fabio Muniz, and probably we'll have some data to submit for publication in the near future. So with that, I will finish thanking all the collaborators involved in this data. So, of course, André Kipnis is the co-PI of all of the projects in the lab. We will have collaborations with Silvia Leão from Unifesp, Roosevelt from University of Federal de Goiás, from Jataí, Cirano Lula from our university, and Vânios Garcia from the University Federal of ABC. Most of the work presented here was done by Fabio Muniz since he was a master's student and now as a postdoc, and also other students are involved that were or are in the laboratory, and they belong to the Rede Goiânia de Pesquisa em Tuberculose. I also need to thank all the sponsoring agencies, CNPq, CAPES, and the federal, the foundation, FAPEG, for their support, and also thank you for listening. So now I'll be glad to answer any questions that we have. Thank you.
Congratulations, Professor. We have 2 questions. Roberta Olmo asked, have you tested iron sulfate or hemming? For Mycobacterium leprae, hemming was able to favor mycobacterial survival, but not iron sulfate, because of the increase of TNF induced the infected macrophages.
That's a good question, Roberta. And actually, this was suggested or asked in one of the thesis on one of my students, and I really have to tell you that we have not tested, because it's really important for us to test how to use hemine as a source of iron, how those bacteria grow. It can grow and use hemine as a source of iron, and we should perform this analysis really soon. I mean, that's not that we will be, but we should be doing. Sorry not to have an answer for that.
Marcia said, excellent presentation. The topic is very interesting. I have a curiosity. There are some studies that show that ATP interferes with mycobacterial iron acquisition mechanisms. Have you studied anything about it? How could this molecule interfere with these mechanisms?
That's a good question, but I'm not a chemistry, and I haven't read anything about it. But you know, in metabolism, you always see a connection between things. So there probably is a connection between ATP and different iron or regulation of expression, but I cannot answer you this question. There are, you know, I think you saw that to cross the membranes, those proteins, either to go, especially when they are internalizing. I showed you ABC transporters and other proteins, even the ESX3 system, they all use ATP. They need a source of energy. Sometimes they use ATP, other times they use the energy, the proton motifs that is already present on the membrane as a source of energy. But specifically how ATP would interfere, I wouldn't be able to answer. I have a question for the students. So I said a lot about iron and all about explanation, and I was curious, and now we are in a pandemic state, and I will not answer, but I would just ask you, do you think there could be any relation of this iron homeostasis from the host and the infection by the virus? And if anybody have read anything, and I just read two articles that came out now because of the pandemic, and they are showing that hepcidin is a marker of COVID severity. So remember that inflammation induces hepcidin expression, and you have inflammation storm in COVID, and now they are, you know, inflammation storm is not only specific for COVID. So many diseases have inflammation storm. So one outcome of the inflammation is the induction of hepcidin and all this influence on iron metabolism. So now people are looking into just not looking at the inflammation as a main outcome and a main cause of disease worsening. So they are looking at these other aspects, and iron availability is one of them. So it's interesting. So for those of you who are in other fields, should if anybody's saying I'm not listening. Anna, are you talking? I'm not listening.
Professora?
Nobody's listening to me?
Oh, now yes.
Yes.
Oh, okay. Okay, I am calling André Bafka to discuss about IL-6, epitidine, and tuberculosis.
Oh, oh, girl. I don't. So do you have a specific question? I don't know anything about iron. I was very curious about the extracellular thing, that MTB, whether MTB could do something with the soluble transferring iron, but Fabio shut me down. So I don't have any questions any longer. No, but I mean, for the IL-6, so the only, yeah, I think the liver response is involved in this hyperinflammatory state, and right, and then you have the transcription regulators that induce all these molecules, heme oxidase 1, I think hepcidin. So I don't have anything to add on this, but it looks like there's a crosstalk between the inflammation, iron consumption, which sometimes I think it has something to do with iron consumption, and that, like the use of iron by mycobacterial tuberculosis, in this case specifically, as a virulence factor. Instead of the nutrition, because it seems to me that that MTB intracellular is exposed to a lot of iron already, right?
There's a recent data that showing how MTB induces ferroptosis, right? And that ferroptosis is induced by high iron content in the macrophage. So although I showed you that the idea is that the host will sequester iron from the pathogen, we can never neglect how mycobacteria is intelligent and have evolved all these 2,000 years in order maybe to circumvent that, and maybe it wants to induce high iron load and cause the damage for the host.
Yeah. So I think the if I can speak a little bit more. So I think the data that I'm trying, I think we also should put in context is that the data when people had anemia and they had TB back in the days, right? Anemia and TB. And then they tried to cure anemia by giving them iron. It seems that the infection was even worse, which goes along with your story that you give more iron, MTB grows better. I'm not sure what kind of assays were performed back in the days. So, but I think there's a correlation with severity of TB and anemia treatment. So I was interested if you guys are doing some experiments with blood, extracellular mycobacteria. Maybe you can have some blood from anemia patients and to play with that.
Good idea, but right. No.
In leprosy, we have a situation that multibacillary patients, they have higher contents of iron inside the macrophages, skin macrophages, and the mycobacteria decreases the expression of ferroportin in skin macrophages. So it's a mechanism used to increase iron inside cells. But we have a limitation because the mycobacterium leprae don't grow in cultures, so we don't have to perform experiments to evaluate the exact role of iron captured by the mycobacteria. But we have found quite the same that is observed in tuberculosis.
Fábio Muniz de Oliveira said, the molecular mechanism of ferroptosis. It's not understand. We see that MTB caused high level of iron, but we did not know how this happened. Isabella Spassadin said, Dr. Kipnis, it is known that there is a relationship between the concentration of intracellular iron and the regulation of the tryptophan pathway. Could you discuss how these specific proteins are able to capture iron could influence directly in this sense?
Isabella, these are tough questions. I don't know the relationship between iron and tryptophan pathway. Fabio, do you know?
No, I don't know. I think the role of iron in the basic metabolism pathway of the cells, like proteins that use iron as a cofactor, can be the relation, but I don't know specifically how the direct influence. I think we do not can think about the iron homeostasis is a direct influence. Iron homeostasis we saw in our lab that when you delete ferritin, we saw the cell wall, the bacteria, have a different kind of phenotype. So I think the iron homeostasis is more complex, and we will not see a direct influence, but you can see how this is going to work in this physiology cell. So you can see the cell wall change, but you not can relate it directly. But the absence of the protein have to be influenced there. So I don't know if it have a direct influence.
Ana Prata said, not just the iron levels, almost all the iron pathway increased.
Mm-hmm.
And about Professor André Kipnis, and lethal oxidation is a good way to understand how the mycobacteria have to get an inflammation during tuberculosis. And we don't understand how this is happening. We know the mycobacterium tuberculosis increase the levels of iron intracellular, but we don't know how molecular mechanism the mycobacterium tuberculosis use for that. It's a good question. We don't know yet if the mycobacterium tuberculosis regulate the host cells or is other mechanism involved there. Do you know or you think about that? Is a good a good way to understand that, or you see something in the transcriptomer that show something like increase the stress oxidative in the cells? Because the final result is a high oxidative stress. It's not so high levels iron, but the final result is a high oxidative stress. So you're not going to see only iron.
Fabio is right, because it's not only iron. Everything else is being modulated inside of the cell or outside of the cell, because when M. tuberculosis, for instance, is inside of the macrophage, he behaves in one way and the cells is producing a lot of things. But when mycobacteria is outside in the granuloma, in the caseous, where there is no cell but subproducts of cell, they produce different molecules or proteins, and the behavior is different, and the metabolic changes are different in those situations. It's not the same. And as Fabio is saying, it's not only iron, but all the inflammatory molecules are upregulated when we have the infection, the macrophage infection.
With respect to macrophage polarization, there are some data regarding this.
Yeah, you wanted to see that, but we did not make that experiment. Professor André did not show in the presentation, but we analyzed the induction of apoptosis by the presence of bacterium, because we think that some way the iron could be influencing in the behavior of macrophage, but we not further for that.
If you vaccinated anemic mice with BCG and challenging with TB, still see protection? Or see more protection?
That's a very interesting question, because if BCG, that it is a mycobacterium bovis, does need iron to survive and to induce immune response that will be protective against tuberculosis, if you don't have a lot of iron to allow the bacteria to duplicate or to induce inflammation, maybe you can have an immune response that it's not so inflammatory, but could be protective. I don't know.
There was a paper that I came across, but I was going to put in the presentation, but I didn't, and because I don't know how to explain very much, but it's basically the amount of iron in the host without infection can skew the inflammatory, the immune response toward Th1 or Th2. So the levels of iron on the host influence, lymphocytes not only differentiation, but also expression of MHCs and other molecules. So when you immunize, so what happens when you vaccinate BCG in an animal that, I don't know if the anemia will reflect a general low level of iron. So you may skew the immune response toward one or the other side. So I don't know. Maybe someone did it. Some Chinese guy did it already.
And I didn't see any correlation between anemia and the different types of T cell population. Have you?
No.
So any more questions, Sarah?
No, professors. We haven't some questions. We haven't no questions.
Sarah, your microphone.
We haven't non-questions in this moment. Finish.
Okay. Thank you.
Okay, then with that, I would like to thank you, André Kipnis, again for this nice presentation, very didactic about the role of iron in the host pathogen interaction. Thank you very much. And thank you all the students, and see you Thursday. at 2 p.m. Bye-bye. Thank you, André. Bye-bye. Thank you, Professor Milton. Thank you, Roberta.
Thank you. Bye-bye.
Bye. Tchau, guys. Tchau, Roberta. Tchau.